Syed Sikander Azam from Quaid-i Azam University. He's a very renowned, that is the associate professor of the university and head of the Department Institute of Bioinformatics. If I'm not forgetting, sir, पूरा नाम शायद यही है कि आगे कुछ है तो आप इसकी तस्दीक कर दीजिए. And he is a person, he's a scientist with a lot of achievements on his behalf, and I just read it out. So that's why you may come to know about the achievements of Dr. Sahib. Give me 1 minute, sir. So this is in fact a short profile of Dr. Sahib. Yes, it is over here. Yes, he has the experience in computational chemistry and molecular dynamics, stimulation of protein complexes to understand their structural and dynamical aspects and role in biological system. Dr. Sikander has accomplished his PhD from University of Innsbruck, Austria in 2009 and postdoc experience from US and Europe. He has produced 6 PhDs and 50 MPhil students along with 85 publications in impact factor journals with more than 200 citations. Furthermore, he is a member of Pakistan Academy of Sciences and has established the Computational Lab at the National Center of Bioinformatics at Quaid-i Azam University. Dr. Sikander has achieved 5 national and 5 international awards, including Research Productivity Award by PCST, Productive Faculty Award by Quaid-i Azam University, and Sampot Fund Award and Ernst Match Follow-up Award. Sorry for the this disturbance. He's in editorial boards of many journals, research journals. He also holds international grants from Pakistan US Funding and International Foundation of Science. So, Dr. Sikander, you are requested to deliver your speech and we'll exhibit the presentations, that PPT. Thank you, sir.
Thank you so much, Dr. Farah, for a nice introduction. As you already informed that I am Dr. Syed Sikander Azam, and I'm thankful for the opportunity you have given to me to speak. So going by slide, I would start my lecture, which is, can you show it on the screen, please?
Sir, it is being shown over here for a while. Wait for a while, for 1 minute only.
Okay.
Yes. here, and you just start your lecture, please.
Okay. Today I'm going to speak about the vaccine and drug designing and the research and strategies behind this. And before that, I would acknowledge for all my work, which has been conducted since long, the International Foundation for Science and PAC US and HEC. Going on to the next slide, please. Here I will tell you about computational biology. Computational biology is actually the field where we use to study the biology. Using the biology, we use the computer techniques in order to run the simulations and to watch the runtime interactions and dynamics of those molecules, which we can express them as a protein. So when you watch it closely, that we have started in 2011, the computational biology lab, and we started with the node technology, which was bread and butter of the bioinformatics at that time. And now we moved to the CPU nodes, and then currently we have acquired the GPU nodes we are funding. GPU is the grand processing unit. So, and we also check them according to the project. We also check our computational findings in experiments as well, but this does not go with each and every project, but it goes from our problem to problem. So today's lecture will go like, if you go to the next slide, please. Yes, today's lecture will be outlined as a computational biology overview and our research highlights, then the vaccine and drug design and the recent trends, then the molecular dynamics for the COVID-19 targets and prevention strategies. Next slide, please. So talking about the computational biology, as I have just introduced it needs the knowledge of different disciplines, and then it is an amalgam of different life sciences approaches and information technology approaches, in which biostatistics, biophysics, and the chemistry of molecules and information of the molecular biology, genetics, these all information and all knowledge is at somewhat extent is useful for computational biologists and for the computational biology, for the field of computational biology. So we use different techniques from these disciplines, and this amalgam actually makes us to do the jobs possible which are actually necessary for the computational biologist. So going to the next slide, please. Well, as the development and application of the data and its analytics and its modeling and the computer simulation of different molecules like proteins and drug targets, and the biology and behavior of and their input in the social system, these all things are actually encompassed in computational biology, which is, of course, the discipline inside the bioinformatics. So going to the next slide, please. Now I'll show you the research, our research area at a glance, which is like we used to. We have a whole group which works in different aspects of computational biology, in which it creates the molecular. It consists of the molecular docking and virtual stewing, and then the molecular dynamic simulation. And we also screen the infectious agents and do the therapeutic intervention inside this. Along with this, we do the algorithm development in bioinformatics, in MD analytics. We have developed our own tools, which I will discuss later, our own analytical methods, which and indigenously developed methods, and also in collaboration with the University of Bristol, UK. So we do have collaborative research as well with international and national collaborators, and these are the areas. The molecular docking, if somebody has not idea, I would explain that fitting a molecule into a 3D space as a raw definition is called docking, so or molecular docking. So in our case, we used to take the target receptor, which is a protein, and if it is overregulated, we used to have a molecule which is going to inhibit it in order to circumvent the problem or ailment or any disease. So, and the molecular dynamics simulation is an important field which actually shows what's happening in the interaction within when we integrate the motion inside it, means what happened under the dynamics. So going to the next slide, please. Well, next, please. So these are the protocol which we follow in our work, and these are the computational parts in which we do the. I have already explained that we do the screenings and dockings and simulation studies. Next, please. So when required, we go to the strain purchase, and we used to check them and verify them in the lab. In our lab also, and also sometimes our collaborators come with their experimental findings, and they want some to deduce some mechanism of that particular their system of interest. We go with that as well. So we also do the algor development, web development, and testing and debugging in programmings, and that is something some specialized part. Next, please. So in just explaining you the research field, I have already explained that we are using the current trend of hybrid molecular docking and virtual screening study. I will define this later, the virtual screening and all that. So the main thing is the computer-aided drug designing. We are actually connected with the computer-aided drug designing, and then we used to we utilize different tools which are actually very useful for the screening and identification of drug targets and vaccine targets. Next, please. This is just to show you that these are a couple of publications which you can see in the molecular docking area. Next, please. These are also some publications. Next, please. And this is the area where our real specialty is, when we see the time-dependent dynamics of certain proteins which are involved, and their role, their changes, their configurational rearrangements, their motions, and their specific function in that motion, and specific move which is responsible for some functionality. So we used to investigate this. Next, please. So these are the research publications which we actually earned from the molecular dynamics simulation side. Next, please. So these are the subtraction of the genome. Means we used to watch the genome of the MDR infectious agents, and there is an approach which is known as the subtractive genomics, in which what we do is that we need the set of proteins which are actually essential for pathogen, but they are absent in human host, so that when it will be this is a new technology which is used in drug designing as well, and this is totally connected with the druggable genome. The druggable genome is a term which is used as like the part of the genome which is druggable, and there are certain filters. There are hundreds of filters which actually perform some analysis, and then they show that okay, this part can be the druggable. So next, please. So in this area, we have certain publications in different journals. Next, please. And this is the algor development area where we used to in-house coding and benchmarking of the relevant programs. Next, please. Yeah, this is the one in the right one is AFD, which is called axial frequency distribution. This is an indigenously developed method in which we watch the protein for the first time in the gradient formation. So their intensity, the intensity of their interactions in different patterns. And this is another method, TUDE, which is actually we have developed with the collaboration with the Institute of Bristol. Next, please. So this is the above graph is showing you the conventional way of explaining this, but in our way we are just showing the gradient information of this interaction, where the intensity of the interaction is higher and where the two body interact, like ligand or amino acid and ligand, if they are interacting, which atoms are actually performing which specific function. Next, please. Well, these are the things about the collaborative research and with the associated procedures. Next, please. Next, please. So these are the work in which the collaboration comes when people are done up with their experiments also, and they want us to deduce that what specific movement, like in one of the work which was in European Journal of Medicinal Chemistry, it was like which ring is actually playing an important part in the under the dynamics and under the simulations, and it is connected. Is it connected with the experiments as well? So we have certain findings in many of our work where the simulation, the movement, the rearrangements and structural flexibility is responsible for different functionality. Next, please. Well, now coming to the students and to the audience, that what is a drug discovery and development is for, and what is a vaccine. I mean, this is not like a mere thing that just drug goes on and it is developed and it is discovered. It's not a very straight away thing. So it needs the whole procedures and whole setup of procedures with different expertise in different steps. So next. So in the first place, what you do is identify the disease, and then you go for this is the traditional development method which I am telling you. This is what happening before the computer aided drug designing and all the virtual screening and molecular dynamics. So you used to isolate each and every protein again and again, and this takes a lot of time. And then you go for finding a drug effective against the disease protein, which another take the 2 to 5 years. So for example, means even if you think about the Panadol, it's before that people were actually consuming salicylic acid, and it took like nearly 30 years to a Panadol to become Panadol. So this is how actually the drug comes on. So then we go to the preclinical testing and the formulation and the scale up things, which is of course taking a lot of time. And this time is actually before the human clinical trials. So it is actually depending on previous steps, that how it goes into the clinical trials of humans, and then of course FDA, which is the drug agency, takes the approval for at least 2 to 3 years. So it's long cycle. It's like 10 to 15 years means to the minimum it's like 18 to 20 to the maximum. Next, please. Next. Yes, now what happened? What happened to this field actually? Why this computer drug designing and this field of computational simulations? Please, yes, on this slide, recent trends. So after identifying disease, the computer speed and capacity have actually paved a way, a new horizon. It's opened a new horizon to every field, and you know that. And that same thing has happened with our field, and genomics, proteomics, and bioinformatics is actually with the information of whole genome and proteome. You go for the high throughput screening, which means that you are going to screen the millions of compounds on the basis of their previous activities and their comparisons. And these comparisons gives you the idea of what is happening inside the molecule. And then you go to the virtual screening to check the whole library of the compounds. And this actually gives rise to the whole branch of chemistry, which is called combinatorial chemistry, where you actually it is the branch of chemistry in which you deal with lots of libraries of compounds. So then you go to the molecular modeling, and here in previous steps, which were like 5 years, it has been reduced because you come with the very specific targets and you check only them. So next, please. Well, same thing is happening with the vaccine. Means you have to isolate, we have to induce the response, and traditionally the vaccine already takes a lot of time, and this is not like, you know, you just for the COVID-19 as well, I would say that this is not like it's just you will go and you will snatch a vaccine from somewhere. It's not going to happen like this. You have to go for each and every step and procedure, which is of course time consuming. While in the traditional way that this vaccine development and discovery, if you see that you have to isolate the peptide and again, and then you go to the like 2 to 5 years, and then you find the construct in 2 to 5 years at least, then you go to the preclinical testing and other steps, which of course takes time. Next, please. While the computer-aided vaccine discovery has changed actually the whole scenario a lot, and what you do in this computer-aided vaccinology or vaccine designing, you have to deal with the reverse vaccinology, where you find there are multiple filters which gives you the vaccine targets quickly and efficiently. So there is a whole procedure behind this, and there is a whole computational experimentations which we take you to the antigenicity finder and then filter. Then you can filter them, like you can design them and you can identify them. Designing that what are the desirable attributes of already ideal vaccines which are actually available in the market. So in comparison with that, you do it with the whole screening step and these steps. And once, like in the modern world, people are going to patent their constructs even computationally, because they can go just directly, like that's happened in meningitis in previous years. So once these constructs come from this side and there is no no hassle in that step, you can go with the identified candidates directly to the synthesis, to the peptide engineers, and you can go for the screening of animal models. So next slide, please. So the interesting fact, which I have already explained, this average design cycle is 6 to 12 years at least, and 10 to 15 years for vaccine, and the success rate is also not so promising in this field. So in, of course, the whole drug designing field. So the average cost is too high with an estimate. Now it's too much, but yeah, this is at least 400 million for the drug and 200 to 500 million US dollars for. So this reverse computer-aided vaccine design and the techniques for the drug and vaccine actually reduce this time and cost to a larger extent. Next, please. Well, in the reverse vaccinology, it helps in the minimizing the costs in terms of human resources and financial resources, and it refines the number of proteins to a large extent, so that the whole steps can be reduced, and the burden can be very less to the synthetic people and to the ultimately to the animal modeling people. Next, please. Well, this is what we have done. And this research article was published in European Journal of Pharmaceutical Sciences. And when the COVID comes, me and my team, we just go ahead with this vaccine designing and this identification thing, and we just carry out this work, and this got international recognition and national recognition as well. Next, please. Can you go to the previous slide, please? Yeah, I should recite this title, which is important. Designing of novel multi-epitope-based vaccine for pandemic coronavirus disease, COVID-19 by Vaccinomics, and probable prevention strategy against avenging zoonotics. So next, please. Well, this study actually presented the comprehensive vaccinomics approach, and we get the protein, whole proteome data from NCBI. and then identified those proteins which can be the ideal target for this to generate an immune response to COVID-19. And we discovered the epitopic regions, the regions which are actually antigenic and have a likelihood of being recognized by the human system with their values. So they were the first. We have done this identification, and then we carry out with those proteins. We have performed the whole procedure. Next, please. Well, the construct have been found. I'm not going to the technical methodology of lots of steps, but the construct was like for the primary immune response, and it was tested. The confirmation of that particular construct was tested by the receptor, by binding with the receptor and checking and validating it again and again, even with the simulations. So next, please. Well, these are the terminologies which is important for the journal audience. Like we used to show that in methodological overview, we just watch all proteins and check them comparatively, that the homology between virus protein, there should be no homology, so that there will be no cross-reactivity, there will be no mouse similar proteins, because when it's going to be checked inside the system, inside the in vitro system, so there is no cross-reactivity to the mouse. And these we came up with the potential vaccine candidates ultimately. Next, please. Well, this is the whole design cycle. This is the whole design cycle which we have performed. This is the whole vaccinology steps, and these are the whole design cycle which we followed. Next, please. Well, this is the ultimate check that this is, of course, this is a state of the art for every computational biologist to check it in that way. Even when we are doing with the experiment, they used to check the stability of these receptors. Actually, TLR3 and TLR4 receptors are important for this pathway, and we have checked that our construct is actually binding it with or not, and we have found promising results with the TLR3 and TLR4 both. Next, please. So this is the exposed part. If you see that this is the whole TLR4 immune receptor, and we docked it and we checked it, and we found it that it is the binding inside the docking and interaction was excellent. Next, please. Well, this is not just binding which is important. We have to go for the simulation to watch the time dependent dynamics of the interaction of this particular construct and the receptor. So in these graphs, we found promising results with both TLR3 and TLR4, and the RMSD, which is actually normally quite high for the constructs. So, but our constructs got some really average RMSD value, which shows that the binding was strong. And even the other RMSF values in the second graph is also under the visible range. And this shows that this binding has been checked again and again. And it is not actually in the whole 100 nanosecond simulation, it is not leaving the active site or leaving the binding site of this particular receptor. This is a very important step, because normally we for peptide, people goes to check it with 20 nanosecond or 30 nanosecond. These nanoseconds are very important. Actually, these nanosecond means previously it takes us the whole 6 months for just 20 or 30, 40 nanoseconds. But now with the development of technologies, and I'm thankful to the Pak-US grant as well, that what we are using the GPU technology, and we used to go with the 100 nanosecond in just a couple of hours, depending on the system or complexity of the system. Next, please. Well, now you can see that with the different colors and the different at 0 nanosecond, at 4.9 nanosecond, at 62 nanosecond, at 100 nanosecond, what is happening, what the colors are changing. But whatever is happening, it's not actually leaving the binding pocket and is tightly formed within that site. Next, please. So this was the initial one, which when it comes to the 0 nanosecond and started exposing to the receptors. Next, please. Well, this is the overall picture of this construct with both of the receptors. So, and you can see in yellow, it has never left the system. And for a peptide to happen like this is very important indicator of its probable vaccine candidate. Next, please. Now I will discuss the second part of this paper, in which I personally have given, or you can say the foremost, I have presented a theory in which facts are present, the theory of retaliation. Means the retaliation from animal-associated viruses or bacteria are actually increasing day by day. And either whatever it is horizontal gene transfer or the transient nature of viral transmission. But if you see the virus, these bacteria in the form of antimicrobial resistance, because one of our whole project is on antimicrobial resistance. So they are the human organ. They have advanced their defense. Organisms have not only increased the capability of offending humans, but they also have developed some capability of advancing their own defense mechanism. So you can check it with the avians, even viruses like infecting hosts. Under this perspective, the avians are much more dominating in this fact. Next slide, please. So this theory of retaliation means that I have recommended that this will play its role in coming future, and for the prevention, for the strategies, we are not going to left with this. The continuous attacks and outbreaks is going to happen in the future, and the dramatic impact of these infectious disease, you already have watched the rays of COVID and corona infections. So this is unexpected. This is just playing havoc with the whole world. Next, please. So in concluding, I would rather say that I will give you the whole conclusion later on about the theory. But in this study, we straight away carrying out our work and prioritize the potential vaccine candidates. And bearing in this mind that they have all the qualities of and being antigenic, conserved, and we have all the qualities which are present in a potential vaccine candidate should be there. And then it was further at the next level, it was checked by the molecular dynamic simulations as well. So these findings actually is presenting a set for a vaccine target directly, and after which you can directly go to the synthesis and screening in appropriate animal models to unravel further effectiveness. Next, please. So meanwhile, the probable prevention in this study for Homo sapien is to avoid becoming a reason for the extinction of various species. I don't know, this will all the people will get it straightforwardly or not, but it is like this. Means we are becoming a reason of extinction of lots of species, and the way we are exploiting animals and sometimes microbes. So these advancements and this theory has to play, the theory of retaliation has to play its role in future. And what we can do is that the whole ecosystem, ecologists in the world, that a balanced ecosystem has to be reinvestigated. We have to watch closely that what we are using, what we are utilizing, and what is in our diet and in our own other procedures in which also, of course, hint towards the environmental pollution and all that stuff. Next, please. These are the references which are already present for this to empower the theory with the facts. Next, please. Yeah, these are some achievements which are already explained. Next, please. Now, this is my group, which is, you can show that we are working on different aspects in Quaid-i Azam University, National Center for Bioinformatics. And next, please. Yeah, this is the whole campaign about awareness on the antibiotic resistance inside the university, in different offices. We conducted that. Next, please. Yeah, last but not least, I would definitely want to acknowledge my international collaborators, Kara Ranajan from Bristol, UK, Klaus Liedl from Innsbruck, Rame from Harvard Medical School, Alex from University of Maryland, and Kate from Bangkok, Chulalongkorn University. So these are the international collaborators, and we are working on different aspects of our work. So these are the national collaborators. I am thankful to Dr. Riaz, Dr. Abbas, and Dr. Imtazuddin from Quaid-i Azam University and Karachi University. I will be thankful to my lab members, Dr. Saad Raza, Dr. Samra Wajid Abbasi, Dr. Asma, Dr. Sajjad, Afifa, Ghulam Abbas, Nausheen, Naila, Rabia, Rosina, Laila, Tayyaba, Saba, Zahra, Namara. And last but not least, I'm really thankful to Honorable Vice Chancellor, Professor Dr. Bushra Mirza, to give me now an opportunity to speak with, and Professor Dr. Farah Khan for organizing this nice lecture. Next, please. And now, thank you for your patience.
Thank you very much, sir, for your valuable lecture. That was really wonderful, and we would very thankful for giving such information which are new in the field of science. Not all in any particular field, but rather all. It covers all the aspects, and it's a sort of collaborative. But I myself, I'm really impressed, and I want to ask you a question. That you're talking about the collaborative research. What do you think? That isn't it sort of a mixed research which is being helped out by your institute or your department or but a lot of different divergent, rather I must say, fields, they would come together and they would make the things which are very different from previous ones. Isn't it like that?
Yeah, actually, if you see the collaboration, it is very important,
Yes.
and this is also very important to how you select your collaborator according to your problems.
Yes.
So normally in the national level, I have seen some, I should not say in words, but means people are like some of their goal is not like just for us sake, for might be for a publication or for something they used to collaborate. But I think the problem is very important. So problem should be global or local, it doesn't matter. But you should know that which important person I have to collaborate with certain problem. So once you're established with this, I think it's a very good chance that you can take your work in a very right direction. So even nationally, we have good people. We can collaborate with them, and we can bring a professional look in collaborations. That will be the only solution for our country, I would say, because one cannot work alone, and even when you are collaborating as a guest, you must be sure that you are not going to be the I should not say the word in words, but you should not going to the parasite on the other person. You should make it like a collaboration. So when this will happen, I think, and as I told you that this vaccine designing or drug designing is not we have different expertise in different steps. You need different expertise.
Exactly. I feel like that. That's wonderful, sir, to have you in our webinar. It was our first webinar over here, and we'll be in touch with you, and we'll be having your opinions ever in doing the things. And thank you very much, sir. Thank you.
Thank you so much. Say my profound regards to everyone and to Dr. Bushra as well. And thank you so much for giving the opportunity.
Thank you, sir. Thank you.